Hello and welcome to another episode of Momento Malum today. Despite the relatively short growing season, Canadian farmers produce a wide range of fruits. The apricot is among these fruit species. For this episode, we'll be covering the genetic diversity of a great many cultivars of apricot from across the world. Let's get started. The first paper cover was made by Dr. Salazar. He analyzed 37 apricot lines from multiple countries using SNP genetic marker technology. These were studied to acquire an estimation of the gene pool within the apricot gene pool. The apricot cultivars tested can be seen on screen right now in the form of a family tree. It was found that these cultivars could be split into two groups. Group 1 is quite large and is further divided into 6 subgroups. Group 2 is smaller. Group 2 consists of heirloom Mediterranean cultivars. Of these are the Spanish cultivars, Marico, Bulida, Currat, and the Italian cultivar Ninfa. Within Group 1, the largest subgroup seem to be closely related to orange-red for subgroup 1 and Hortic for subgroup number 4. Another paper that covers this topic was done by Dr. Batnini. This paper involved the use of physical traits and SSR marker technology in order to analyze the gene pool of many apricot lines. The lines analyzed were 42 Tunisian lines and 30 Spanish lines. It was found that these cultivars cluster into 3 genetically distinct groups. Group 1 contains 16 lines from the Spanish collection. This includes the 7 cultivars Currat, Goldrich, Mirlo Rojo, Valorange, Marchiania, Celine, and Orange Red, as well as 9 experimental lines. Group 2 contained 29 lines that are further divided into 3 subgroups labeled A, B, and C. A consisted of 9 Tunisian lines, B consists of 4 Tunisian lines and 4 Spanish lines, and C contains 10 Spanish lines and 2 Tunisian lines. All the listed cultivars can be seen on screen at this time. Group 3 likewise has multiple subgroups, also labeled A, B, and C. A consists of the descendants of the cultivar Bergeren. B consists of cultivars from southern Tunisia, and C consists of the remaining Tunisian cultivars. This process also found that the Spanish breeding program has incorporated a greater deal of the variation within the apricot genome than the heirloom and experimental cultivars of Tunisia. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. Still, another paper was written on the same topic by Dr. Pedryc using SSR markers. This experiment involved a genetic analysis of 77 apricot cultivars originating from 5 different regions. These can be seen on screen right now. It was found that these cultivars broadly group into clusters based on their region of origin. Group 1 consists of cultivars that have other related species in their pedigree and are not pure-blooded apricots. Group 2 consists of mostly Middle European and exotic cultivars with Middle European progenitors. Many of the North American cultivars have some traces of Middle European pedigree within their lineage. The third group consists of 20 out of the 23 Chinese cultivars. Interestingly, Apricot, a North American plumpox virus resistant cultivar, clustered with the Chinese genotypes. This might be explained by the fact that Apricot has perfection and scout among its ancestors, both of which are of Chinese pedigree. This may indicate that plumpox resistance may be found primarily in Chinese cultivars. The Mediterranean, North American, and Central Asian cultivars seem to be a composite of the above groups, derived from the mingling of these genetic lineages. We'll conclude with a paper by Dr. Lee, who would likewise use SSR markers. to analyze the gene pool of the cultivated Chinese apricots. It would analyze 76 common cultivars grown in China, which include 32 native Chinese cultivars, 20 that have their origins in Central Asia, 9 from Europe and North America, 2 from the Near East, 5 used for their edible seeds, and 8 plum apricot hybrids. It was found that all these groups cluster together genetically speaking, i.e. all Chinese cultivars form one genetic group, as did all the other groups. The common model of plum domestication is that China and Central Asia have the oldest apricot populations and by proxy the most genetically diverse populations. This study likewise supported this model. And that about covers everything. Thank you for watching.